Okay, so we have started talking about the classification of lipids, and in this regard, the first thing we have done was to talk about fatty acids. In this lecture, we are going to talk about the other remaining types of lipids. So the next type we are going to talk about that is called as triacylglycerols. These lipids are also called as triglycerides. Triglycerides are made up of glycerol and fatty acids. You can easily remember the structure of glycerol. Here you can see this is a 3-carbon hydrocarbon. It's an alkane called propane. If a hydroxyl group is attached to each of these 3 carbons, that molecule is glycerol. Glycerol contains hydroxyl groups on it. These hydroxyl groups can undergo esterification with the carboxyl groups of fatty acids. So, if one hydroxyl of glycerol is esterified with a fatty acid, that is called as monoacyl glycerol. When two hydroxyls undergo condensation with two fatty acids, the resulting structure is a diacyl glycerol. And when all three hydroxyls of glycerol undergo this reaction with three fatty acid molecules, the resulting molecule is called as triacylglycerol. It is sometimes also called as triglyceride. And on this next slide, you can see this figure from the book. This shows the structure of glycerol here. And when glycerol undergoes esterification with these three fatty acids, it produces triacylglycerol. Triacylglycerol are also called as triglycerides. These are non-polar substances. They are insoluble in water, and they are produced through an esterification reaction between glycerol and fatty acids. So this is a generalized representation showing any three fatty acids esterified to glycerol. In this figure, you can see three different types of fatty acids attached to glycerol. The first one, shown in red, is palmitic acid. It is a 16-carbon fatty acid. The second one shown here is an 18-carbon fatty acid, linoleic acid. Linoleic acid is an unsaturated fatty acid. So you can see that between carbon 9 and 10 and carbon 12 and 13, there are two double bonds. The third one shown here is a saturated fatty acid, which is stearic acid. It is also an 18-carbon fatty acid. So therefore, the name of this triacylglycerol is 1-palmitoyl, 2-linoleal, 3-stearoyl glycerol. Animals and plants store excess of these molecules in form of a mixture of triacylglycerols. Fats and oils, they contain many different types of triacylglycerols in which different types of fatty acids are conjugated to glycerol. Plants, as we have talked about this earlier, the oils of the plants, they are rich in unsaturated fatty acids. So therefore, their melting temperature is low. For this reason, oils are liquid at room temperature. Triacylglycerols are an important reservoir of energy. This figure shown here represents a micrograph of adipocytes. Adipocytes are fat-storing cells. The fats are stored in the cells of the body in form of triglycerides. One important function of fats we have talked about is that it acts as an insulation. Fats present beneath the skin is important for maintaining the body temperature. In addition, fats are also important for other processes as well. One such example are the sperm whales. These are huge animals They contain a large amount of fats, a mixture of fats present in their head, which is called as spermaceti. It is a mixture of various different types of fats which contain several types of fatty acids in them. It is typically important because when whales are close to the surface of water, the temperature is higher. At that temperature, the fats present in the head, they are liquid and hence the density is low. But when the whale is diving deep in the ocean, there is a large amount of pressure which is exerted. So in order to counter that buoyant pressure, when the whales dive, the temperature also drops. This decrease in temperature results in turning the fats into solid. So the transition temperature is such that close to 31 degrees centigrade, these fats are going to turn into a solid. Hence, that results in increase in the density. This increase in the density helps in countering the pressure, the buoyant pressure, which is exerted at great depths when the whale is diving. When the whale again comes closer to the surface, the temperature again increases and the fats, they are again going to turn into a fluid. Therefore, this transition of spermaceti fats from solid to liquid and liquid to solid helps the whales in diving and swimming. The next type of lipids are called as glycerophospholipids. They are named so because they contain phosphate attached to glycerols. So they are condensation products of phosphoric acid and glycerol esters. As shown in part A of this figure, this portion is glycerol. The hydroxyls of glycerol are represented in red color. The hydroxyl on the third carbon of glycerol undergoes a reaction with phosphoric acid. So this phosphate is attached to glycerol and hence this compound is glycerol phosphate. These hydroxyls of carbon 1 and carbon 2, they undergo a condensation reaction with the carboxyl group of fatty acids. Therefore, the resulting molecule is called as glycerophospholipid. Several different types of other polar groups which are represented by X here are also attached to this phosphate molecule. Depending upon the nature of the group attached, these phospholipids are named. This part forms the polar head group of phospholipids, and the two fatty acids attached on carbon 1 and carbon 2, they form the so-called hydrophobic tails of the phospholipid molecules. Two layers of phospholipids assemble in the membrane, hence they constitute the lipid bilayer. This table here shows different types of polar head groups which are attached to glycerophospholipids. So this is the generalized structure of glycerophospholipids with a polar group attached to this phosphate represented by an X. X could be a variety of different types of polar groups. If no other group is attached, that is, X is a hydrogen atom, so it is called as phosphatidic acid. If the polar group attached here is ethanolamine, the structure is shown here, then the resulting phospholipid is called as phosphatidyl ethanolamine. If choline is attached, then the resulting phospholipid is called as phosphatidyl choline. It is easy to remember the structure of ethanolamine and choline if you know the structure of ethanol. Ethanol is a two-carbon compound and it contains a hydroxyl group on one of the carbons. If you substitute the hydroxyl group of ethanol with an amino group, the resulting compound is ethanolamine. So this two carbon compound having an amino group on it is ethanolamine. And if you can recall the structure of ethanolamine, it is easy to remember the structure of choline. So if you substitute these three hydrogens attached to nitrogen by methyl groups, the resulting molecule is choline. Phosphatidylcholine is also known as lecithin. It's a common name of phosphatidylcholine. Lecithin is commonly used as an emulsifier in several of the food products. For example, if you would look at the ingredients of any given chocolate, you would find soy lecithin or some other lecithin which is added as an emulsifier. Phosphatidylcholines, they generally are called as lecithin. The next type of polar group attached is serine. Serine is an amino acid. The structure of serine is shown here. This is a central carbon, which is called as alpha carbon. All amino acids contain a carboxyl group attached to their alpha carbon and an amino group as well. So there's a common structure to all amino acids. They contain an alpha carbon, a carboxyl group, and an amino group. Amino acids differ from each other in terms of their side chain. This part is called as the side chain. The side chain of serine contains a hydroxymethyl group on it. So a one carbon unit is a methyl group, and when a hydroxyl is attached to it, it is called as hydroxymethyl. So it is very easy to remember the structure of serine. There is an alpha carbon to which a carboxyl group and an amino group is attached, and the side chain is hydroxymethyl. This hydroxyl group of serine undergoes condensation with the phosphate of phosphatidic acid, as a result of which this entire part of serine is attached as a polar head group to this phospholipid. The resulting phospholipid produced is called as phosphatidylserine. The next one shown here is inositol. This is the structure of inositol represented here. You can remember inositol from the carbohydrate derivatives. Inositol is a product produced by the reduction of glucose. Inositol contains 6 carbons. It's a cyclohexane, and on each of the carbons, one hydroxyl group is attached. So this is cyclohexane hexanol. That means it is a cyclohexane which contains 6 hydroxyl groups on each of the carbon. One of the hydroxyl groups undergoes a reaction with phosphatidic acid, and therefore this inositol is attached to phosphatidic acid, thus producing phosphatidyl inositol. You would remember that we commented about phosphatidyl inositol earlier as well. Through some signaling events, this inositol is released from the membrane phospholipids in form of inositol triphosphate. This inositol triphosphate then moves through the cytoplasm of the cell, and it binds to specific proteins. In turn, it is important for regulating the flux of calcium in the cell. And by regulating the flux of calcium, it is useful in controlling many different types of phenomena. For example, it is important for its secretion from the cell. It is important for regulating the transport of vesicles in cell and several other processes. So, phosphatidyl inositol has a very important role in cell signaling. Some phospholipids contain glycerol as their polar head group. Such lipids are called as phosphatidylglycerol. Some complex lipids, they contain a phosphatidylglycerol as their polar head group. That means such molecules, they will be having two phosphatidylglycerol molecules. They're called as diphosphatidylglycerol and commonly called as cardiolipins. You are required to remember the structure of phosphatidic acid and all other glycerophospholipids. On this next slide, you see a representation of a glycerophospholipid. The lipid shown here is phosphatidylcholine. This part shown here is glycerol. These are the two fatty acids attached to carbon 1 and carbon 2. You can see that the fatty acid attached to carbon 2 is an unsaturated fatty acid. This is carbon 3 to which the phosphate group is attached, and this part is choline. So this is phosphatidylcholine. This is another representation of the molecule shown here. This is a space-filling representation. This is fatty acid attached to carbon number 1, and this is a fatty acid attached to carbon number 2. So you can, in this structure, clearly see the kink which is produced because of the double bond. This part here is the phosphate, and the choline part is represented here. So this is the structural representation of phosphatidylcholine. Certain types of glycerophospholipids have common names. For example, phosphatidylcholines, they are called as lecithins. Lecithins are widely used as emulsifiers in different types of food products. Diphosphatidylglycerols are also called as cardiolipins. They get their name because they were first isolated from heart. Some different types of lipids, they contain an ether bond rather than a typical ester bond. For example, they're called as plasmalogens. Another example of a lipid containing an ether linkage is platelet activating factor. This is produced by certain types of lymphocytes, and it is important for the aggregation of platelets. Aggregation of platelets is needed for the process of blood clotting. The platelet activating factor is important for several other functions as well. Here is a diagrammatic representation of plasmalogen and platelet activating factor. You can see here that on carbon 1, there is an ether linkage. This is glycerol, the phosphate, and the polar head group. This is the structure of platelet activating factor shown here. So here you can see that this part is glycerol onto which the lipid is attached through an ether linkage. So generally, glycerophospholipids, they contain an ester bond, but some types of phospholipids, they contain an ether linkage as well. The next type of lipids, they are called as sphingolipids. These are derived from sphingosine or dihydrosphingosine. The structure of sphingosine is represented here. These type of lipids do not contain glycerol in them. Rather, they have this nitrogen-containing compound which is called as sphingosine or dihydrosphingosine as represented in this structure. This is a representation of a typical sphingolipid here. So this part shown here is sphingosine to which a fatty acid is attached. Different types of polar head groups are attached here represented as X. These polar head groups are shown here. If X is a hydrogen atom, then the sphingolipid is called as a ceramide. If phosphocholine is attached to sphingosine, it is called as sphingomyelin. If glucose is attached, these are called as glucocerebroside. Several different other types of oligosaccharides can also be attached. These are called as gangliosides. This figure shows the structure of gangliosides. This is for your information only. You are not required to remember the structure of gangliosides. This figure shows the structure of phosphatidylcholine, which is a glycerophospholipid, and sphingomyelin. They both contain choline as their polar head group. So you can see that sphingomyelin and glycerophospholipids, they have similarity in their structure, and they both are present in the membranes of cell. They constitute the lipid bilayer of the cell membrane. Carbohydrates, when attached to sphingolipids, are called as glycosphingolipids. Here is another important example of glycophospholipids which determine the blood groups. The ABO blood group antigens are glycosphingolipids. Here you can see this part is the sphingosine to which a fatty acid is attached, and then this oligosaccharide is attached to sphingosine. This typical arrangement of oligosaccharide is present in case of O antigen. This is sometimes called as the base oligosaccharide. If N-acetyl galactosamine is attached to this base oligosaccharide, it gives rise to A antigen. And if galactose is attached to this base oligosaccharide, the antigen is called as B-type antigen. ABO blood groups arise because of this typical type of oligosaccharides present on these glycosphingolipids. They are present on the membranes of red blood cells. The enzymes which carry out the breakdown of phospholipids are called as phospholipases. There are several different types of phospholipases depending upon the site of action on which they carry out the breakdown of phospholipids. Phospholipases A1 are those which carry out the hydrolysis of the ester bond between carbon 1 of glycerol and the fatty acid. Phospholipase A2 are those enzymes which carry out the hydrolysis of the ester bond attached to carbon number 2. The enzyme which breaks down the phosphorus bond is called as phospholipase C. If a phospholipase C breaks down the bond, this phosphate is released from the phospholipid. Phospholipase D are those enzymes which break down on this position. Therefore, the phosphate group is not released with the polar head group. We have previously talked about phosphatidyl inositol, the structure of which is shown here. This polar head group attached here is inositol. Through cell signaling, the enzyme phospholipase C is activated. This enzyme carries out the hydrolysis of this phosphate bond and releases inositol triphosphate. This inositol triphosphate is transferred to the cytoplasm of the cell, where it acts as a second messenger. It binds to proteins and eventually it results in the opening of calcium channels on the endoplasmic reticulum, thereby releasing calcium into the cytoplasm of the cell. As we have talked about this earlier, this release of calcium is important for several important physiological functions. For example, cellular secretion, movement of cytoplasmic vesicles, and so on. So phospholipases are those enzymes which break down phospholipids, and they're classified based upon the bond which they hydrolyze. And the last type of lipids which we are going to discuss collectively are called as sterols. Cholesterol is one such example of sterols. Sterols, they contain a cyclic structure. They contain this fused ring system which is called as cyclopentano perhydrophenanthrene. Phenanthrene is a fused ring made up of benzenes. When three benzenes are fused with each other, they give rise to this ring structure called as phenanthrene. So this is the same phenanthrene ring shown here, and this is a cyclopentane, a five-cornered ring attached to phenanthrene. So this is the basic structure of these compounds, which are called as sterols, and cholesterol is one of those compounds. So therefore, this is the basic ring structure called as cyclopentano perhydrophenanthrene. This is a diagrammatic representation of the structure of cholesterol. Here you can see the fused ring system of cholesterol. This is the side chain. Cholesterol, in addition, also contains a hydroxyl group. Cholesterol largely is a non-polar molecule except this hydroxyl group which is present here, sometimes which is called as the polar head group. This is the surface representation of this molecule, so you can see that it is a planar, rigid ring structure. Cholesterol is an important constituent of the animal cell membranes. Cholesterol is a planar structure and a rigid ring system. If inserted into the membrane, it decreases membrane fluidity. One of the ways animal cells regulate their fluidity of the membrane is by regulating the amount of cholesterol in their membranes. Higher is the amount of cholesterol in the membrane, less is the fluidity of that lipid bilayer, and vice versa. Another very important function of cholesterol is that it is a precursor to steroid hormones. There is a variety of different types of steroid hormones which are produced by different parts of the body. These include the hormones which regulate carbohydrate metabolism as well as the sex hormones. Plants also have a little amount of cholesterol in their membranes, but cholesterol is not a predominant sterol found in plants. Rather, they contain some other type of sterols in their cells. Cholesterol, in many cases, undergoes esterification. This is the molecule of cholesterol, and this is the polar hydroxyl group present here. The hydroxyl group of cholesterol can undergo esterification with fatty acids. Some cholesterol exists as free cholesterol, but most of the cholesterol exists in form of esters. Cholesterol has several important functions in the body. For example, it is a precursor to bile salts. Bile salts are present in bile, which is produced by the liver and is important for assimilation of fats. Cholesterol is also a precursor to vitamin D. By exposure to UV light, cholesterol undergoes a conformational change and is turned into a precursor to vitamin D, which later through chemical reactions is converted into the active form of vitamin D. Vitamin D is needed for the dietary absorption of minerals from the gut. In turn, it is important for the development and maintenance of bones. Cholesterol is also a precursor to all of these different types of steroid hormones and is an important constituent of the cell membranes. It is important for regulating the fluidity of the membranes. So indeed, it is a very important molecule. Some cholesterol is present inside the cells and some of it is present in circulation. The cells of the body also synthesize cholesterol. Another way of acquiring cholesterol is through the diet. So because of these important functions, cholesterol is an essential substance needed by the body. Excess of circulating levels of cholesterol is indeed bad for your health. Excess of cholesterol and several other substances in circulation can accumulate in blood vessels and can cause blockage. Cholesterol is transported by special type of proteins which are called as lipoproteins. These two types of lipoproteins which transport cholesterol are called as high density lipoprotein or HDL and low density lipoproteins or LDL. LDL, low density lipoprotein, transports cholesterol from the liver to other parts of the body. Sometimes LDL cholesterol is also called as bad cholesterol because it results in deposition of cholesterol as well as in circulation. HDL cholesterol is sometimes called as the good cholesterol because HDL, the high density lipoproteins, they transport cholesterol from other parts of the body to the liver, that is from extrahepatic tissue to the hepatic tissue. In the liver, cholesterol is metabolized and is converted into other substances, including the bile salts. Regulation of blood cholesterol level is important. If the blood cholesterol level exceeds the permissible range, then this can result in accumulation of cholesterol and therefore can cause blockage of blood vessels. People suffering from increase in their circulatory cholesterol level are treated by a type of drugs called as statins. Statins are those drugs which inhibit an enzyme which is involved in the biosynthesis of cholesterol. It is a key enzyme called HMG-CoA reductase. So statins are those drugs which target and inhibit that enzyme, thereby suppressing cholesterol biosynthesis. So once there was this fellow who went to a shop and bought a pack of oil. Then he went to the shopkeeper and said, "Hey, where's my free gift?" The shopkeeper looked at the items and said, "Sir, there are no free gifts with these items." So the fellow pointed out to the label of that bottle which said, "cholesterol free." If you are unable to get this joke, please replay. So nonetheless, cholesterol is an important substance for the body, although an excess of cholesterol is bad, but cholesterol is needed for many important functions being carried out in the cells of the body. So this is all about the various different classes of lipids. In the next session, we will look at the role of lipids with respect to the membranes.